Hello, everyone, and welcome to today's webinar, Advancing the Science and Accessibility of Human Milk for a World Without NEC. We are thrilled to have you join us. Throughout the presentation, you can submit your text questions to our presenters by typing them into the questions pane of the control panel, which is located at the bottom right of your screen. Feel free to send in your questions anytime. We'll collect them and address them during the Q&A session at the end of today's presentation. To maximize your webinar experience, you can participate using other toolbar panes, including handouts, chats, event handouts, including a certificate of attendance with continuing education credit, can be found by clicking the handout section of the toolbar. And if you experience any technical issues, please click on the help button on the lower left of your screen. Please note that today's webinar is being recorded. And after the event, you will receive a follow-up email with the link to view the recording. And without further ado, I would like to now introduce today's speakers. Dr. Lars Bodde is Professor of Pediatrics at the University of California, San Diego, the Lars and Rosenquist Foundation Endowed Chair of Collaborative Human Milk Research, Founding Director of the Larson Rosenquist Foundation Mother Milk Infant Center of Research Excellence, Mommy Corps, and the founding director of the Human Milk Institute, HMI, the only academic institute in the world that is dedicated to all aspects of human milk. And Jennifer Canvasser, who founded the NEC Society after the tragic loss of her son Micah. Through Jennifer's leadership, the NEC Society has become the world's leading NEC-focused nonprofit. Jennifer graduated from UCLA, earned her master's from the University of Southern California, and is a PhD student at UC Davis. Jennifer has led 3 PCORI Engagement Awards, as well as the Chan Zuckerberg Initiative's Rare as One Network and patient-partnered research collaboration. She has completed 15 marathons, donated over 5,000 ounces of breast milk, and authored the storybook Forever Our Little One. Thank you again for joining us. I'm going to turn it over to Dr. Bodde to get us started.
Thank you very much. 1 second. And here we go. All right, good morning, everyone. Thank you for having me. I think this is a unique seminar or webinar format because you have those two very different speakers presenting on the same topic and coming from 2 very different perspectives. The biggest difference is that I have never run a marathon before, whereas Jennifer is running them like they're going out of style. But really the difference here is that I'm looking at necrotizing enterocolitis purely from a scientific research perspective and see how we can utilize human milk and the knowledge around human milk to end necrotizing enterocolitis. And then Jennifer being affected by necrotizing enterocolitis personally, and really taking that energy and that passion to create the NEC Society, and together with clinicians, researchers, educators, and everyone else trying to end necrotizing enterocolitis for good. So you'll hear these 2 different perspectives, and I'll kick off with talking about the powers of human milk, and specifically the powers of milk oligosaccharides, which is our research focus here in my lab at UC San Diego. And I'll show you why human milk oligosaccharides might be part of the solution when we talk about necrotizing enterocolitis. So I'll take a few of the slides here from the NEC Society that Jennifer had founded to introduce what NEC is and why we're actually talking about it. So NEC, or necrotizing enterocolitis, is a devastating intestinal disease that primarily affects premature infants, can also affect term babies, especially when there's other underlying medical conditions. And a few statistics here. 1 out of 10 very low birth weight babies develop NEC. Sometimes these numbers are even higher. NEC is the most common cause of death for extremely preterm infants between 2 weeks and 2 months of age. And stunningly, 1 baby dies from NEC every single day in the United States alone. So that's 1 baby that shouldn't be dying of NEC, and we need to do something about it to eliminate NEC from this world. Okay, so I'll give you a couple of other introduction slides here about necrotizing enterocolitis, just to bring all aspects of the scientific numbers and everything. So this is also NEC. This is a baby affected by NEC. You see this severely extended belly. If you go a little bit deeper, here's figures here from a paper that was published in the New England Journal of Medicine a while ago. Again, you see that extended belly. If that baby has surgery and you see the intestine here being necrotic, and there's this extended air in the abdominal wall that is very characteristic for necrotizing enterocolitis. So, as introduced in the beginning, it is still one of the most common and devastating intestinal disorders in preterm infants. About 5 to 10% of all very low birth weight infants develop this often deadly intestinal disorders. There's a high mortality rate, and even if you the survivors often are faced with long-term nutritional and neurological complications. We don't really know how NEC comes to be, so the etiology and pathogenesis is multifactorial and still poorly understood. It happens very rapidly. A baby can be fine in the evening, and the next day, it's unrecognizable. Very progression, and we have currently limited treatment options. So bringing this all together, it's of course most desirable to prevent necrotizing enterocolitis altogether before it strikes. And that's where human milk comes in. We see that infants who receive human milk are at a 6 to 10-fold risk to develop necrotizing enterocolitis. Those are huge differences. And the question is, of course, what is in human milk that is so powerful to protect preterm babies from necrotizing enterocolitis? So let's take a look at human milk here for a few slides. What is in human milk that makes it so powerful? Well, certainly there's water as the main ingredient, if you want. That is, of course, critical. But then there are carbohydrates, mainly lactose as the milk sugar, oligosaccharides that I will be talking about for the remainder of this presentation. There are lipids that are packaged in the milk white globule with highly active components surrounding the triglyceride core. We have proteins, peptides. These proteins can be proteases, and they help and aid in the digestion of other proteins as they enter the infant. And then there's many components. We have cells in human milk, bacteria in human milk. Milk is not sterile. There are vitamins and minerals, of course, hormones, and then many, many other bioactive components. And truth is, for many of these components, we don't even know what they are at this point. So if you do an untargeted metabolomics approach on human milk, there's so many different components that we still haven't identified that are in human milk and are potentially bioactive. So we also know that human milk is more than just a list of ingredients. So while we can identify what is in human milk, it's way more than just a list of those things. And paper here in Science a few years ago where we talk about human milk as a biological system that is this mother milk and trial, which in itself sits in this socioeconomic behavioral context that is very different depending on where you are in the world and in which setting you are. So if you look at this triad here, again, there are socioeconomic factors, cultural factors, behavioral factors, environmental factors that determine where this triad sits. And that includes, in our case here, the preterm infant in the NICU being very different, of course, than the term infant, let's say, in sub-Saharan Africa. And there are fixed and modifiable factors on the maternal side as well as on the infant side that we need to take into consideration. It's not just the composition of breast milk or a one-size-fits-all approach. And this, in fact, changes over time. It changes over the course of lactation. In fact, it changes not only within a day, but within a feed. Foremilk and hindmilk are different in composition. So there's a lot of dynamics going on in this triad, and depending on where it sits in its context. And that, of course, is very important when we talk about human milk, let's say for necrotizing enterocolitis. So let's break this down into just macromolecules in human milk. So simplifying this entire system that I just tried to make more complicated, the macromolecules in human milk, the fat, the milk sugar, lactose, and then there are oligosaccharides, up to 15 grams per liter, which often exceeds the amount of total protein. If we compare that to other mammals, especially those that we use to make infant formula, when we look at the milk of cows, for example, there's about 100 to 1,000-fold less oligosaccharides. And these structures are in composition very different than they are in human milk. And since cow milk or dairy products are the basis for most infant formula, the oligosaccharide composition is very different. And we're only now starting to add some of these oligosaccharides that are synthetically made into infant formula to compensate for some of this difference. So again, if we look at this question here, what is in human milk that makes it so powerful to prevent necrotizing enterocolitis, and we massive difference in oligosaccharides between human milk and everything else, including infant formula, our hypothesis was, well, maybe it's the oligosaccharides that contribute to some of these benefits we see when it comes to necrotizing enterocolitis. So a few words about human milk oligosaccharides. What are actually these human milk oligosaccharides, or HMOs? They're complex sugars, and these sugars are made of 5 simple building blocks, or monosaccharides, that are glucose, galactose, N-acetylglucosamine, fucose, and sialic acid. And depending on how we put these 5 building blocks together, we get different oligosaccharides that have different structures as well. And I often compare this to Lego bricks, where if you had an arsenal of different Lego bricks and you put them together in different ways, you would get different things. So you either get this little car here, or you get a little plane, and you would agree that planes and cars do different things. And that's the same for oligosaccharides as well. They're not interchangeable. Every different oligosaccharide does different things. Structure determines their function. So I'm showing you a few oligosaccharides here, and the important ones will pop up at the end, where I'm showing you the blueprint, as we call it, in the left upper corner. And you see that all these oligosaccharides start with lactose at the reducing end, and then they get extended with disaccharide units in different linkages. They can be branched, and they can be further extended every time with a disaccharide, so 2 monosaccharide building blocks. And this is only using 3 building blocks so far, this glucose, galactose, and N-acetylglucosamine. And you see there's a huge difference already in their structure. They can be like branched and clumped, or very linear spaghetti-like structures. And these oligosaccharides we call the neutral non-fucosylated oligosaccharides. And non-fucosylated means there must be something that's fucosylated, and that's the next step. That is addition of the 4th building block, fucose, in different linkages. So fucose can be added to lactose at the backbone here, and then you get 2-fucosylactose and 3-fucosylactose. And I'm highlighting here 2-fucosylactose 2-FL because it is one of the most abundant HMOs in human milk of most women. And you'll see in a few slides that there are some women that don't make this oligosaccharide. And this oligosaccharide has become quite important, as it is added to some infant formula now, sometimes together with other oligosaccharides. But there's also data showing that this oligosaccharide, at least in tissue culture, has some impact on necrotizing enterocolitis. And I'll show you at the very end that, unfortunately, we don't have any evidence that that is relevant to humans, but we'll go through this as we get closer towards the end. So keep that in mind. We can also add fucose here to the different growing chains and different linkages, and extend and add more than 1 fucose to it if we want to. And all these oligosaccharides now we call the neutral fucosylated oligosaccharides because they contain fucose. Now, next and last step is the addition of sialic acid. So sialic acid contains a carboxyl group with a negative charge, so it changes the characteristics of these oligosaccharides completely. We once again can add sialic acid to either lactose, and that gives us 3 and 6-sial-lactose, or we add it to the growing chain, and we get different structural isomers here where we add sialic acid to in different linkages to these growing backbones. And you see in the right corner down here, DSLNT has 2 sialic acids, and we'll get back to this particular oligosaccharides in a minute. So these are the acidic or sialated oligosaccharides because they carry this acidic group, carboxyl group, that contributes the negative charge. And then last but not least, we have oligosaccharides that contain both of those modifications, so sialic acid and fucose, and those then are the acidic or sialated and fucosylated oligosaccharides. Most importantly here is we have about 150 to 200 different oligosaccharides that have been identified by structure. And not every person makes the same oligosaccharides. It varies between different people quite a bit, and it changes over the course of lactation. Again, speaking to this dynamic mother milk infant triad, and we'll get to that in a second as well. So here are the main research focus areas currently that are around human milk oligosaccharides. What are the maternal drivers that determine what oligosaccharides are made in a given individual at a given time? Most importantly, what are their benefits for infant health and development? And lastly, certainly for necrotizing enterocolitis, can we utilize that knowledge to develop new therapeutics or biosolutions to treat diseases like necrotizing enterocolitis or prevent them before this? A few words about maternal drivers. Is data from the Canadian child cohort, where in this case we analyzed about 1,200 different milk samples from different subjects at around the same time of lactation. And you can see how different the oligosaccharide composition looks like, different colors being different oligosaccharides. And then you see concentration on the y-axis. On the very left, very low concentrations. On the very right, very high concentrations. And some of these oligosaccharides vary multiple folds between individuals. And then it is not only that the total concentrations vary, but it is their relative abundance as well. So if you took all these oligosaccharides to 100%, you see that different individuals just make very different oligosaccharide patterns. This is a slightly different view of how this looks like for oligosaccharides collected from about 10,000 different subjects. And I think we're up to 40,000 subjects by now, but I haven't updated the graphics here. And what you see here in this three-dimensional space is every dot in this space represents the HMO composition, the total composition, in a given sample. The closer those milk samples are together in this three-dimensional space, the more similar the oligosaccharide composition. And the further apart they are, the more dissimilar or different the oligosaccharide compositions. And you see there's like these two different clouds here, right? A bigger cloud on the right and a smaller cloud on the left. And we can show that that is actually strongly determined by genetics. So if we do a genome-wide association studies on these oligosaccharides and compare different polymorphisms with oligosaccharide expression, then you see there's a very strong signal standing out here with a p-value over log 100. So that's 0.000 and 100 zeros until you get to the 1. And we see there's two different arms on the chromosome that are affected. If you just look at the stronger signal here, you find that there is a gene called FU2 that is associated with this massive difference. And that is encoding an enzyme, is a fucosyl transferase, so it transfers fucose to your oligosaccharide backbone in this alpha-1,2 linkage. And if you have this SNP meaning you have one base pair in your genetic code that's slightly different, you can't make this enzyme. This enzyme is no longer active, and you're not adding fucose in this alpha-1,2 linkage to your oligosaccharides. So think about that. You have one single base pair in your genome that is slightly different, and it changes your milk oligosaccharide composition dramatically. Completely. This is not just one oligosaccharide different, it's almost all oligosaccharides different. It also affects glycoproteins, glycolipids, and many other things that are glycosylated in your milk sample. So I always find that fascinating, that there is one single base pair that can just change your composition completely. So we call this the secretors and the non-secretors. Doesn't mean that the non-secretors can't secrete milk. It also doesn't mean that they can't secrete oligosaccharides. It just means that they secrete slightly different oligosaccharides, or actually different oligosaccharides, dramatically different. And the biggest difference is that the secretors have 2FL as their most abundant oligosaccharide, whereas 2FL is near absent in the non-secretors. This is different in different parts of the world. So it is clearly driven by genetics and different geographies. However, if you look at the same individual, massive change in saccharide composition between individuals, but within the same individual, over a short period of time, the oligosaccharide composition is remarkably consistent. So you see on the top here, taking a milk sample every single day for a week, hardly any difference in oligosaccharide composition. You go higher in resolution, you take a sample every hour, barely any difference in the composition here over the course of the day. But very different if you take this data from two different subjects. So bottom and top are two different subjects. And this is absolutely important for research because it tells us that if we take a sample independent of the time of the day, it doesn't matter if it's fore milk or hind milk. It doesn't matter if you take it on a Monday or on a Thursday. You get a fairly representative sample for your oligosaccharide composition in that window, let's say a week or two. And that is very important for something that I'm going to show you later when we start to associate oligosaccharide composition to necrotizing enterocolitis incidents. So with that in mind, this is not the case for many other milk components that change quite a bit over the course of lactation or over a week, so sometimes over a day or a feed. If we look at longer trajectories of oligosaccharides, and this is 2 years postpartum here plotted out, you see there are some saccharides that are changing. Most oligosaccharides decline in concentration. 6-lactose is going down within a month or two by multiple fold. Other oligosaccharides like 3-fucosyl-lactose increase in concentration, also dramatically, to the point where in some women they become the most dominant oligosaccharide after a year of lactation. So for some strange reason, 3-fucosyl-lactose becomes way more dominant in the later stages of lactation than in the beginning. We don't know why that is, but we've observed this in multiple different cohorts around the world. Okay, so much for the genetic drivers. So we're still trying to entangle this. We still don't know exactly how human milk oligosaccharides are synthesized in the human membrane gland. We're working on that. And then there are other factors like maternal nutrition, physical activity, use of probiotics, maternal health states, or gestational diabetes, for example, use of certain therapeutics and drugs that alter human milk oligosaccharide composition to another extent. So clearly, genetics are the main driver of oligosaccharide composition. So that's just an intro to the maternal drivers. What's more important here is, of course, what do these oligosaccharides do for infant health and development? And when do we get to necrotizing enterocolitis? Because that's really the main topic of this presentation. So let's take a look at what these oligosaccharides do. Let's take a look at the mechanism. Before we get there, let's take a look at what these oligosaccharides, where these oligosaccharides go once they're ingested. We know that they are fairly resistant to the low pH in the stomach, to the pancreatic and brush border enzymes. About 1% is absorbed in the small intestine, and you can measure it in the urine. And the rest is either degraded by bacteria in the colon or excreted intact, and you find it in the fecal sample when you measure it. So the question is, well, what do these oligosaccharides do? How come that they're just degraded by bacteria, or 1% being absorbed and potentially having systemic effect? Is it a lot? Is it a very small amount? So what exactly are these oligosaccharides do? And if we look at this diagram here, where we map out oligosaccharides on the top, potential good bacteria in green on the left, a potential pathogen here in this violet color in purple on the right, and then intestinal epithelial cells and the immune cells on the bottom, we know that these oligosaccharides are prebiotics. So they serve a certain type of bacteria that can metabolize these oligosaccharides, use them as an energy source. These bacteria have a benefit over other bacteria and potentially create a benefit for the infant. So this is like the typical definition of a prebiotic. You feed bacteria, and then these bacteria do all the magic. But we clearly see that HMOs are more than just food for bugs. So it's way too simple to think that all these 150 different structures are made simply to feed some individual bacteria. It's way more complicated than that. And one thing that they do is they actually do the opposite. They serve as antimicrobials. They stop the growth of certain bacteria and in some cases even kill certain bacteria. At the same time, they serve as anti-adhesives. We know that certain bacteria need to attach to the intestinal epithelial wall, and oligosaccharides are serving as anti-adhesives or anti-attachment factors that block certain bacteria from attaching. And if you can't attach, you're just simply getting washed out without causing any harm. So that's another effect, how oligosaccharides can shape microbial communities without serving as a prebiotic. And then there's other effects that are microbiome independent. So oligosaccharides have a direct effect on epithelial cells in the intestine. And that can change intestinal epithelial programming completely, which then might have an indirect effect on microbes in the intestine. We also see that oligosaccharides have an effect on immune cells in the intestine. And that might also then have an effect on microbial communities in the gut. So there's this strong interplay of microbiome dependent and independent effects that all together shape not only microbial communities in the gut, but overall the health of the intestine and the gut of the baby. And then I said that some of these oligosaccharides, or not some, but about 1% of the oligosaccharide gets absorbed and makes it all the way into systemic circulation. So in addition to the local effects in the guts, we also see that there are systemic effects of these oligosaccharides, specifically on immune cells, on macrophages, for example. We see some effects on liver cells, adipocytes, and others as well, in a concentration range that is indeed relevant to these absorption rates. And the truth is, we are probably currently here. We still have a lot to discover, and with new tools becoming available, actually new oligosaccharides becoming available, we'll be able to uncover more and more of these effects that are currently under the surface and not visible to us. Okay, so let's talk about the actual research and then necrotizing, because that's what we're here for. Oligosaccharide research currently looks at pathogens, for example, diarrheal disease, respiratory infections. How can oligosaccharides help in that case? We look at oligosaccharides for infant growth and body composition measures. We look at oligosaccharides when it comes to the immune system, allergies, asthma, all those kinds of conditions. And we also look to the brain. Do these oligosaccharides have an impact on brain development, on neurological outcomes? So these are all research areas that are currently in the focus for human milk oligosaccharides, but truly is we're looking at necrotizing enterocolitis here. And that brings us to the important part of this presentation. Where do oligosaccharides play a role in necrotizing enterocolitis? So here's the hypothesis. Do oligosaccharides play a role in preventing NEC? So first hypothesis is that HMOs in general contribute to the protection from NEC. But the second is that the inter-individual variation in human milk oligosaccharide composition, so the changes in oligosaccharide composition between different individuals, explains why some infants still develop NEC despite receiving human milk. And very important. Like I said in the beginning, not all oligosaccharides do the same, but also not all individuals make the same oligosaccharides in the same concentrations. So we'll test these two hypotheses. So how do we test this? Oligosaccharides don't grow on trees. We can't just do an intervention study and feed them to babies and then see what happens. We start very simple with an animal experiment. So we take oligosaccharides, isolate them from human milk, from pooled donor human milk, to get the entire garden variety of different oligosaccharides. And then we test this out in an animal model of necrotizing enterocolitis here in neonatal rats that we feed ourselves with infant formula that either contains, well, not infant formula, but formula that is adapted to this rat model here, that either contains oligosaccharides or doesn't. And then we see what happens to those pups. Do they develop NEC-like symptoms, and do they survive longer? You see here how the formula is dosed with this syringe here, 200 microliters a piece, with and without oligosaccharides. And here's the first data that we got from this study. And you see, just by looking at survival, so this is loss of animals due to NEC-like symptoms in the DF or dam fed control. So these are the animals that were left with mom. Mom fed the babies just the way it should be. All animals survived the first 96 hours, not a problem. If we feed just formula without oligosaccharides, you see we lose about 25% of the animals. If we add human milk oligosaccharides, so these oligosaccharides that we isolate from human milk ourselves, to that very same formula, the survival rate goes up to almost the dam fed control. So we lost 1 out of the 20 animals here. That to us was absolutely stunning to see that just by adding oligosaccharides, everything else was the same. We had massive improvement in survival. If we then look at the intestines, you see that the FF is the formula-fed group. Intestine looks way more bloody, a little bit distended. So there are NEC-like symptoms that we observe just macroscopically. We then go in and section the intestines and stain them for microscopy and score this by pathological scores. Again, you see down here that the dam fed control on the pathology score, it's almost 0. There were a couple of animals that had a slightly higher score, and 0 being a completely normal intestine. before being completely destroyed intestine. The formula-fed group, though, had animals all along the spectrum. Some animals were fairly healthy, and some animals' intestines looked completely destroyed. Again, if we add human milk oligosaccharides, the very same formula, the pathology scores dropped almost down to the dam fed control. Massive, significant difference. If we take what's seen here on the very right, galacto-oligosaccharides, so these are oligosaccharides that are not in human milk, but still are oligosaccharides, there was no effect. So the effect is specific to human milk oligosaccharides. So next we wanted to see, is this relevant to all human milk oligosaccharides, or are there structure-specific differences? Are there specific oligosaccharides that are responsible for this effect? And we did this by what we call a multi-dimensional chromatography approach, where we separate the oligosaccharides first by charge, so depending on how many sialic acids they have. We found that the oligosaccharides with 2 sialic acids were most efficacious. And then we looked, okay, what is in this fraction? What kind of oligosaccharides are in there? Can we identify which oligosaccharides are responsible? We then, in the second dimension, separated the oligosaccharides by size. So remember that backbone structure in the end can be very small or can be very extended. And that's what we looked for here. And we found that there is one specific oligosaccharide with 2 sialic acid molecules and a specific length that is most effective when it comes to reducing the pathology scores here. We then looked at what this oligosaccharide looks like by structure, and this takes about 9 months. You just can't look at it with a microscope or something like that, or however you want to. You need some sophisticated methods to look at glycan structure. And when we did that, we found that this oligosaccharide that is most protective in the animal model looks like this. So it has an L backbone, these glucose acids. So DSLNT stands for for disiallyl, so 2 sialic acids, lactose, which is the backbone. Now, we presented this data at the conference way back. You see this data is fairly old already. And neonatologists would immediately tell us, congratulations, you've got an animal sick and then you cured it. But this is not NEC, has nothing to do with what we see in the preterm infant. So please go back and show us that this actually translates to the human preterm infant as well. Again, this is about 2014, I would say, 2015, when we presented this data first. And still, oligosaccharides not growing on trees. We don't have any handle on getting enough DSLNT to do an intervention study. So we had to do another intermediate approach here. And that was to look at a mother-infant cohort, where we specifically recruited moms and their very low birth weight infants, collected milk samples, analyzed them for oligosaccharide composition, and then measured which oligosaccharide is potentially associated with NEC incidence. And I'm going to show that for our first cohort here in a second. So the hypothesis here is that infants who develop necrotizing enterocolitis received milk with less of this potentially protective DSLNT than infants who do not develop NEC. So in this case, we only recruit infants that receive exclusively human milk. So no formula feeding in this study. And this was a study that was done in 5 different centers in North America, with San Diego being the prime. We recruited between 2012 and 2014, specifically very low birth infant under 1,500 grams. And then we matched infants that received developed necrotizing enterocolitis with control infants that did not develop NEC. And this is how this looks like. So on the top, you see a few characteristics on the left, and then from 1 to 28 is the first 28 days of life. When you see this N in a red box, that means that baby had necrotizing enterocolitis at that stage. And we have milk samples when you see the numbers in the green boxes. So these are milk samples that we have available to analyze. And then we match that with 5 to 6 different infants, about the same gestational age, same birth weight, same delivery mode, same race or ethnicity, and where we had milk samples at around the same time. So we measured all these milk samples, about 2,000 milk samples for this particular study that we analyzed. And then we plotted here over the first 28 days postpartum, the concentrations of oligosaccharides here for all oligosaccharides combined. So the sum of all oligosaccharides. And the different colors here on the plot are the gray dots in the background are all the match controls where babies did not develop necrotizing enterocolitis. The white, yellow, and red dots on here are milk samples or oligosaccharide compositions in milk samples of infants that later developed necrotizing enterocolitis Bell stage 1 in white and 2 and 3 in yellow and red. And if you aggregate all this data, you see there's no difference in total oligosaccharide composition between the controls and between those infants that later developed NEC. And that was not our hypothesis that there would be any difference. The hypothesis was that there is a difference in this potentially protective HMO called DSLNT. And that's what you see on the top of the figure here. So if we now plot DSLNT concentrations over time, you see the gray dots, so the controls are all over the place, but the milk samples, so the DSLNT concentrations in milk samples that were given to babies that developed specifically Bell stage 2 and 3 NEC are at the very low spectrum of DSLNT concentrations. And if you aggregate that, you see that that is significant. We then did some additional modeling of this data, and here you see, for example, in these red and blue dots, these are individual milk samples for the different cases that we looked at over the course of 28 days. The more red the color, the lower the DSLNT concentration in that sample compared to controls. So you see for DSLNT, every single milk sample here had lower DSLNT concentrations compared to controls. And if you look at other oligosaccharides, so LSTB, for example, just lacks that terminal sialic acid. There's no correlation, no clear association between this oligosaccharide and the development of NEC. And the same is true for other oligosaccharides too. Now, that shows that there is a significant association with low DSLNT levels in milk given to babies being associated with a higher risk of that baby to develop necrotizing enterocolitis. And you can actually go through these thresholds here where you say if the DSLNT concentration is beyond or above a certain threshold, then the baby is very unlikely to develop NEC. If it's below, then there's other decision points here for other oligosaccharides that then tell us whether a baby is more or less likely to develop NEC. So this is very important for us, of course, to be able to screen mom's own milk and to develop technologies to screen donor milk products. And we'll get to that at the very end. Okay, so we then had two more cohorts, one in South Africa, and I'm not showing here, but one in the UK, where we worked with the biorepository in the UK, and Chris Stewart is the collaborator here, where they had now 33 NEC cases and 37 controls. We analyzed the oligosaccharides and saw, yeah, there is a difference clearly if we look just at the composition overall. And if we look at individual oligosaccharides, is there differences? Yes, there is. There's one oligosaccharide that stands out as being significantly associated with NEC, and that is once again DSLNT. So we have three different cohorts now: North America, South Africa, and the UK, where we see that DSLNT is associated with necrotizing enterocolitis. And the thresholds actually matched pretty well. What we see in the cohort here in the UK matched very well at what we saw in the cohort in North America. So it's just a little bit off by a few micrograms. Okay, so we have a match. We have a match where the different cohort data, the associations actually match the causalities in the animal model. So that helps us to further inform our clinical studies to now take DSLNT to the clinic, and also to continue to study the mechanism. Now, a couple of words about the mechanism. This gets very scientific now, but I think it's very important as we're developing these new therapeutics. The hypothesis originally was that DSLNT is metabolized by microbial communities. So it served as a prebiotic that I introduced earlier. And then those microbial communities benefit the host and protect from necrotizing enterocolitis, right? So it's an indirect effect where DSLNT does something to the microbes, and then the microbes do something to the host, and that benefits or protects from NEC. So we tested this hypothesis, a very simple experiment here where we took bacterial communities, microbial, from either NEC patients or from patients that did not have NEC, so babies again matched. And what you see here is that we take all these different oligosaccharides from 2FL on the left to DSLNH on the right. And the more the red these boxes are, the more those microbial communities metabolize these different oligosaccharides. And what really stands out here is when we look at DSLNT, that the NEC cases or the microbial communities in the NEC cases metabolize most of the DSLNT, whereas the controls actually spare DSLNT and do not metabolize it. So it's exactly the opposite of what we had thought in our hypothesis. So DSLNT is metabolized by microbial communities isolated from NEC patients, but not from control patients. So for us, that was a huge surprise. And it also turns out that if you do studies on intestinal organoids and you treat it with different oligosaccharides, DSLNT happens to be the most active HMO to drive differential gene expression. So our hypothesis now is that it's not used as a prebiotic, but it's actually doing something directly on the host. And we need to make sure that the microbial communities do not utilize DSLNT, but instead that DSLNT is protected, is spared to be able to have a direct effect on the host. And that really is part of our strategy when we look at solutions. So what we really want is not just DSLNT or just having a broadband probiotic that nibbles on all different oligosaccharides, but really a symbiotic, a smart symbiotic, where the probiotic spares the oligosaccharide DSLNT so that DSLNT can do something else. So summarizing this, we've seen that HMOs contribute to the protection from NEC, and also that there is an inter-individual variation in HMO composition, and depending what oligosaccharides are fed to babies, it puts them in a lower or higher risk to develop NEC. And that brings us, of course, to the solution. So we talked about the problem, but what's the solution? So can we take this knowledge and develop new solutions to prevent NEC altogether? And of course, there is an opportunity to develop therapeutics. Can we take DSLNT, make it available synthetically, preterm infant, and hope that the NEC incidence is going down? Beautifully, oligosaccharides, human milk oligosaccharides, are indeed now becoming available for a while already, either because they're chemically synthesized or there's a chemoenzymatic approach to make them available, but mostly currently they are synthesized in what's called bioengineered microbes in these huge fermenters. So there is bacteria, again, bacteria sometimes being the culprit, but in this case actually being the helper. Feed these bacteria simple sugars and they spit out your more complex oligosaccharides. And that's currently happening. That's happening for oligosaccharides like LNT that are made in these large fermenters and are currently added to infant formula. For DSLNT, unfortunately, the story is a bit different. DSLNT is more complex and it carries this internal sialic acid that makes it very difficult to synthesize at large scale. So if we wanted to give DSLNT to a preterm baby in the first 28 days of life, so over the first 4 weeks, we calculated it would cost us currently about $900,000 per baby to do that. And fortunately, NIH just awarded a grant, a small business grant, to a startup company out of Cornell called Zamtronics, where we're part of. And their goal is to bring down the costs of DSLNT through new synthetic approaches from $900,000 to hopefully somewhere around $5. So if that's successful, then we certainly have a path forward. Currently, this is a feasible path. But there's also a different opportunity. There's an opportunity to develop new diagnostics. So if we look at these thresholds again, right, the thresholds that we identified in the different cohorts where we need to have a certain amount of DSLNT to be protective against necrotizing enterocolitis, then is that something that we strive to measure in human milk, either at point of care or in milk banks, to be above that threshold to give optimal protection? Now, when it comes to the milk bank, here we looked at the distribution of DSLNT concentrations in about 200 active qualified milk donors around the US. And you can easily see how huge the spread is of DSLNT concentrations between different donors. So these that eventually end up in a pool of donor milk. And the NEC preventing threshold is about down here. So 23% of the currently qualified donors that we surveyed below that threshold. And even if we pooled these milk samples together and we happen to pool the wrong donors together, we would still be below the NEC preventing threshold. So how do we know what kind of DSLNT concentrations we have in individual donors and then eventually in the pooled product? Is there technology we can use to measure this other than the mass spec or HPLC that's currently in labs? And to do that, we teamed up here with a professor in the Department of Electrical and Computer Engineering who does for a living, he builds small technologies to measure things, small. So we teamed up with Drew Hall and developed this point-of-care technology to test for human milk oligosaccharides. In this case, we measured two focusolactose concentrations because here you have this all or nothing, the secretors and non-secretors that you can measure. And it turns out this is a small chip. It's like smaller than your cell phone for sure. It's Bluetooth enabled. The results go straight onto your iPhone or other smartphone that you have. And without going through the different technologies here, it not can determine secretors not black and white, but it accurately measures the concentrations of 2-fucosolactose within 20. So we're now taking this technology, change it around a little bit because it's not that for DSLNT, oligosaccharide that we're looking for. And we're currently working, and we have a prototype already in place that can measure concentrations at point of care as well. So that's the two main opportunities, building new therapeutics, but also looking for diagnostics. So all in all, we're trying to harness the power of human milk to improve and save preterm infant lives. Currently, NEC is the most common cause of death, and we need to change that. We know that human milk is part of the solution, and oligosaccharides might be one of the drivers. So in summary, speaking about oligosaccharides and their link to NEC, HMOs are the third most abundant component of human milk. The composition is greatly in individuals, which is mostly driven by genetics. Within the same individual, it's remarkably constant over a period of time, which is extremely important for research purposes. So if you think about those association studies, if oligosaccharide composition would go up and down over a short period of time, it would be very difficult to make those conclusions and drive those associations. But since they're fairly consistent over a short period of time, we can do that. They do change over the long course of life, so over a year or two. And that is important, maybe not so important for necrotizing enterocolitis, although we have to keep in mind here that DSLNT concentrations also change over the longer course of lactation. And if we're using donor milk, the donor milk often is not coming from the first few weeks of lactation. They're coming when lactation is established, few months out, and the oligosaccharide composition of those donors might be very different than the oligosaccharide composition of what the baby needs in the first couple of years of life. We know that HMOs remain undigested by the host, reach distal parts of the gastrointestinal tract, and there they can serve as prebiotics, but also serve as antimicrobials, antiadhesives, epithelial cell modulators, and immune cell modulators. And we believe for necrotizing enterocolitis, it's more so, or it's less so the prebiotic and more so the cell modulating effects. And they also may have systemic effects. When it comes to human milk oligosaccharides, they're not interchangeably. So DSLNT seems to be the most powerful oligosaccharide, both in the animal study, but also in the 3 independent cohorts. So DSLNT reduces NEC-like symptoms and survival in the rat model, or improves survival in the rat model. And, very important, it's not one or the other, it's both. In association studies, we also found that lower NEC incidence is associated with higher concentrations of this oligosaccharide. Now, other human milk oligosaccharides, like 2-fucosolactose, may affect NEC-relevant pathways in tissue culture, sometimes are shown to reduce NEC-like symptoms in animal models, but, and I need to highlight that here, there is currently no evidence that these other oligosaccharides, other than DSLNT, are associated with reduced NEC in human preterm infants. So just because this oligosaccharide, like 2FL, is available, and it's much cheaper than DSLNT, doesn't mean that it becomes part of the solution. Right? It's nicely to sell this, but it might be more complicated than that. So a better understanding of the role of human milk and its bioactives, like HMOs in general, and DSLNT in particular, creates unique and powerful opportunities to develop new therapeutics based on the natural and safe template human milk, and develop new diagnostics to inform healthcare milk banks to provide the right baby with the right milk at the right time. And this is a phrase that we coined at one of our milk banking workshops here at the Human Milk Institute a few years ago. Okay, so take a look at these numbers one more time that I introduced this topic with. This needs to end. We need to change it. we need to work together to build a world without NEC. And from the research perspective, human milk plays a huge role in this, and oligosaccharides in particular. So I invite you to join us for the next Human Milk Institute Symposium, March 10 to 12. If you've been there, you know this sells out very quickly. So if you see the announcement coming up, sign up. We do always have a section on nutrition support for the preterm infant in every single symposium. And specifically, Jennifer, who's going to talk about NEC from a very different perspective, was one of our guest speakers. And we're partnering with the NEC Society to bring human milk and human milk research to end necrotizing enterocolitis. So I'll end there and hand it over to Jennifer. Thank you.
Thank you, Lars. I'm going to go ahead and bring up my slides. Welcome, everyone. I am so honored to be here with you all. So my presentation is going to be sharing my perspective from a bereaved mother who has been traumatized by this very devastating disease. And as introduced at the beginning of the webinar, I founded and I now am the executive director of the Necrotizing Enterocolitis Society. And these are my twins, Micah and Zachary, who you will be hearing more about in just a moment. Here's what we're going to cover together over the next 45 minutes. I'm going to share why I'm here and what drives my work at the NEC Society, the urgency of addressing necrotizing enterocolitis, the gaps in NEC research, what we can do and how we can partner with patient families, and what you all can do today to help us build a world without NEC. I'm going to start by actually sharing my very personal story and journey with NEC. This is my husband, Noah, and when we found out we were expecting twin boys, we were ecstatic and overjoyed. I've always thought of myself as a very healthy person. I did everything that I thought I should to keep my babies healthy and closer to full term, but they were born at 27 weeks gestation. They each weighed 2 pounds. And I had never been in the neonatal intensive care unit before. I knew nothing about necrotizing enterocolitis or about pumping. I had never even seen a breast pump before I had an emergency C-section. But within, the matter of days and weeks of our babies being in the neonatal intensive care unit, Noah and I learned how to be NICU parents. We learned how to provide skin-to-skin care for our babies. I was able to build up my milk supply for the twins. And by the time they were 6 weeks old, they had doubled their birth weight. They weighed about 5 pounds. And they were on kind of what I thought the track to home. They were beautiful and thriving. This is Micah, just hours before he developed necrotizing enterocolitis. And this was after he developed NEC. And you can see that he's back on the ventilator, back on pressors, and was critically ill within really a matter of hours. He went from being stable and healthy and thriving and what I thought would be approaching discharge soon to being critically ill and being taken to the operating room where he had multiple bowel resections take place. And you can see that this photo or video of Micah and Zachary together, Micah is extremely fluid overloaded. And that's because after he developed necrotizing enterocolitis, he went into renal failure as a result of just being so sick from NEC. But we were very lucky. We were at a large academic institution with a really incredible nephrology team. And so they transferred Micah from the neonatal intensive care unit to the pediatric intensive care unit. And they placed him on continuous renal replacement therapy. And it pulled the fluid off. And practically miraculously, Micah opened his eyes and he smiled at us. And it was really the start of many dreams coming true. I thought that after Micah was diagnosed with NEC and he was taken to the OR multiple times, I was terrified of losing him then. And so to see the nephrology team have success removing the fluid, it was really incredible. We were exhausted, as you might imagine, caring for a very critically sick child. And then thankfully his twin brother, Zachary, was healthy and was discharged home. And so we spent every day back in the pediatric intensive care unit with Micah. My husband at that time was in residency. And so it was really a challenging experience for him to balance being in residency and having a child in the pediatric care unit, the ICU. But we managed to do okay. And at this point, you could see Micah is doing well. He had a lot of complications from necrotizing enterocolitis, but we knew that he would be coming home and that we were going to give him everything that he needed to thrive. He did have short bowel syndrome because, as I mentioned, he had multiple pieces of his bowel removed. He had to go back and forth to the OR to remove necrotic bowel. His kidneys never recovered from the initial infection with NEC because he became so sick so fast. His kidneys never recovered, so he was reliant on hemodialysis and continuous renal replacement therapy. But you can see he was beautiful and thriving, and I knew that Micah was going to be okay. I knew that he was going to be coming home. And even though his kidneys never recovered, I planned to be his kidney donor. I was going to donate one of my kidneys to Micah. And again, we were going to give this baby everything he needed so that he could thrive and live the life that he needed to be with our family. And so these are just some videos and photos of us showering Micah with love, still in the PICU, still working on getting Micah home. I often would joke with Micah's care team that I was just going to leave the hospital. I didn't care if they weren't ready to discharge him, that I was just going to take my baby and walk home because we lived only a mile from the hospital. They didn't think my jokes were too funny, but thankfully they did discharge Micah and we did get to bring him home, and bringing Micah home was really a dream come true for our family. And while we were at home together, he got to meet more of our family members who couldn't come to the hospital to meet him. He got to meet neighbors. We went on walks around the neighborhood, and we get to do normal family things like have dinner at home together. And here they are the twins. They were about 10 months old in this video. And you can see Micah still had a feeding tube. He was still on oxygen, but again, he was going to get everything he needed from us to thrive. But tragically, he developed pneumonia. He had a lot of complications from necrotizing enterocolitis. He had liver disease, lung disease, end-stage renal disease, short bowel syndrome. And so he ended up back in the pediatric intensive care unit. But even then, I looked into his beautiful eyes and I saw his bright smile. And I knew that Micah was going to be okay. I knew that Micah was going to come back home. But I was wrong. Micah never came back home. And just before the twins' first birthday, I held Micah in my arms as he left this earth. And I have spent every day since then working desperately to keep his smile in my life. We held the most beautiful life celebration for Micah, and I was so thankful that many people from his care team joined us. It meant the world to us to have Micah's surgeons, nurses, dietitians, everyone there with us celebrating Micah's beautiful smiles and life. And this is many members of Micah's care team. And I'm very thankful that many of these people have remained with our family and have continued to contribute to the NEC Society. And it is devastating. It will always be devastating. And I have been able to keep Micah in my life by helping us build a world without necrotizing enterocolitis. Micah had every reason not to smile when he was in the NICU and PICU. He was poked and operated on and had more painful procedures than most of us will ever have in our lifetime. And still he smiled. And so I hold on to all of Micah's magical smiles, and I use them to propel my work at the NEC Society and to inspire all of us to do what we can to help prevent necrotizing enterocolitis from devastating families just like ours. So I want to highlight a few key takeaways from my family's story. The first thing I want to make sure that we all understand is that mother's milk does not eliminate the risk of NEC. I hate that that is a truth, but it is a truth. You can see Micah's twin today is thriving and healthy and wonderful. And thank goodness he never developed necrotizing enterocolitis. He was protected against it because they both received my milk. So mother's milk is the most potent, powerful thing that we have to reduce the risk of NEC right now, but it does not eliminate the risk of NEC. Tragically, many babies, babies still today who receive their mother's milk still continue to be diagnosed and die from this disease. So we need to do more. We need to get more babies their mother's milk, protective donor milk, and still we need to do more to find what we need to build a world without this devastating disease. For all families, not just for bereaved families, but families whose babies survive this disease, it is often a life-altering diagnosis. It affects and changes families for a lifetime. And not just the parents or not just the patient who's affected and diagnosed, but the siblings, the grandparents, the cousins, everyone who loves that child is changed for the rest of their life after a NEC diagnosis. Trust and rapport in the neonatal intensive care unit and the pediatric intensive care unit are essential. We need to take time to build relationships, nurture relationships between patient families and the clinicians who are providing the care. And I really insist that we change the attitude around necrotizing enterocolitis. And I am seeing a shift because when Micah was diagnosed with NEC, his clinicians shrugged their shoulders at me and said, well, this is just what happens to some babies who are born preterm. NEC is just something that happens. There's not really much we can do about it. And that is not true. NEC is not inevitable. We can and we must do more for our babies and families to protect them from this devastating diagnosis. It is not inevitable. We need to make it preventable and eliminate the risk of NEC because babies are counting on us. So that's why I'm here. That's why I have poured my heart and soul into building this organization, the NEC Society, over the last 10 years. And so now I want to talk a little bit more about the urgency of necrotizing enterocolitis. These beautiful families and babies that you see on your screen, they have all been tragically bereaved from NEC. All of these babies have died from this disease. Right now, as we speak, babies and families are being devastated by NEC. It often develops suddenly and moves aggressively. Tragically, a lot of the families who are diagnosed with NEC, they only have hours or maybe days before their child tragically dies from this disease. I feel very lucky, even though we tragically lost our son, and I will hate that every day for the rest of my life. I'm so thankful that we had nearly a year with our baby, that we got to bring him home, and we got to create a lifetime of memories with Micah, because many families don't have that because NEC is so aggressive and it comes on so suddenly. It blindsides not just the families who don't know about the disease, but even the clinicians, the experts that are taking care of these babies are often blindsided by NEC because it comes on so quickly, often at a time when babies have made it through their initial, oftentimes most challenging time in early days in the NICU, right? When the babies are at their earliest gestation and they're learning how to breathe, right? These babies oftentimes will get through that initial really difficult period, and it will affect them like Micah when they're 6 weeks old, 7 weeks old. And so weeks after, months after they've overcome the most, challenging time of being born preterm, then they're diagnosed with NEC. So it's often comes on very suddenly, very aggressively. And so we do need to focus on prevention. And I think it's highlighted really powerfully here between these two photos. These photos were taken hours apart. And so you could see Micah before he developed necrotizing enterocolitis. He weighed over 5 pounds. He was beginning to nurse at the breast. He was thriving and healthy, and he should be today. Within hours, he had underwent multiple bowel resections. He was extremely fluid overloaded, couldn't move his body because he had so much fluid on board, and was kind of at death's doorsteps. It was absolutely horrifying and just torture to watch Micah go through this and to be helpless. And I'll say that it's not just the families who feel helpless. The clinicians are at the bedside caring for them often feel helpless when this is happening to one of their patients, and they don't have the tools that we need to prevent this from happening and to treat babies like Micah. As I mentioned, our babies cannot wait. We need a world without NEC today. We need to do more right now to prevent NEC, to get life-saving human milk to more babies, to increase and optimize the accessibility of protective donor milk, because as we speak, our babies are being diagnosed with and dying from NEC. All of these babies on your screen here have been diagnosed with necrotizing enterocolitis. Some of them have tragically passed away. Many of them are living with life-altering long-term complications. And I will I want to say here, I might say this again later, but I want to make sure we recognize that the complications from NEC don't end when a family leaves the NICU. This is a disease that affects families often for a lifetime. So it doesn't end, the complications from NEC don't end at the end of antibiotics. They don't end when the baby has a successful bowel resection. It doesn't end when these babies leave the NICU. It doesn't end when they're toddlers or into childhood. We have a lot of families and patients in our community who are experiencing the long-term complications of NEC well into adulthood, into their 40s. And it doesn't end when they leave the NICU. It continues for years after the original initial NEC diagnosis. So that's why I'm here. That is, you can see the urgency of preventing NEC, of building a world without NEC. So let's talk a little bit more about the gaps. What causes this disease? We actually don't know, and that's not acceptable. We need to figure out why some babies develop NEC, why some babies, like Micah's twin, do not, thank goodness, develop NEC. It's complex. We're not going to find one single cause. We're not going to have one single treatment. It's multifactorial, and we need much more research to understand why and how NEC develops so that we can stop it from happening. We have a lot of research initiatives within the NEC Society and our partners trying to figure this out. We have the NEC Biorepository led by Dr. Misty Good, who is leading this effort. And I encourage you all to check out our website if you're curious, like what type of research is going on to figure out why and how NEC happens. This is Micah's twin brother, Zachary. And he was about 4 years old when he asked, "Mommy, why didn't I develop NEC and die like Micah?" And one day I hope that we can answer Zachary's question, because right now we don't know why. We don't know why one twin develops NEC and tragically dies when the other one is a thriving, beautiful child today. And I will always believe that Micah should be too. I will always believe that Micah's NEC was preventable, that we could have prevented it from happening so that Micah would be like Zachary is today. And that's what really drives me. We also don't know why sometimes NEC affects term babies. And so it typically affects babies that are born prematurely in the NICU, but that's not always true. And I would say our patient families have really elevated this truth, because when I founded the NEC Society, I was under the impression that it was an exclusive disease that only affected premature infants. And our patient families have helped me learn that that is not true. Many term babies develop NEC, especially if they have an underlying condition like a congenital heart defect. And so we still need to understand and learn and do more research about how do we also not just affect our preemie infants from NEC, but our term babies, our heart babies, our cardiac babies. How do we ensure that no infant ever has to face the devastation of NEC? We went through an elaborate process at the NEC Society to identify some of the key NEC research priorities and gaps. And so you can see many of them here. We need to optimize human milk and lactation support. We need many more tools to improve the diagnosis of the disease so that babies are diagnosed much earlier and sooner. We need to have a better understanding and way to treat the long-term complications of NEC and to support families who are navigating this devastation. We need to optimize communication in the NICU. You can see it's a pretty long list of research priorities and gaps that need to be addressed in terms of ensuring that we are prioritizing research for this disease. So now I want to talk a little bit more about how we can partner with patient families. I do believe that our partnership with patient families are one of the most powerful pathways that we have to building a world without NEC. And that's because there's no one more invested in a world without NEC than our families who are living through this devastation. And I think we all need to start with recognizing that every single baby in the neonatal intensive care unit needs mother's own milk or pasteurized donor human milk. That should be the standard of care everywhere. There should not be any exception to that rule. Every infant that is born prematurely and therefore at risk of necrotizing enterocolitis needs the protection from mother's milk and donor milk, period. Every family in the neonatal intensive care unit who has an infant at risk of this disease needs to know about it. Most families today, despite all of the efforts that we are taking at the NEC Society, Most families continue to learn about NEC when their child is being diagnosed. That is not acceptable. Families need to know about NEC, about the protective factors, about the risk factors. They need to know that their child, because they were born preterm, they're at risk of NEC. And they need to know so that they are empowered to advocate for their child, and they know about the life-saving power of human milk. And so when should we begin talking about NEC? I've heard from a lot of clinicians, well, we don't want to overwhelm families with too much information. We don't want to scare families. Families are already overwhelmed. We're already scared. Having information is not more overwhelming. It's actually empowering, right? It allows us to have a roadmap of what we can expect, how we can navigate, how we can show up for our child during this really terrifying time. And so families should learn about necrotizing enterocolitis when we're talking to them about the life-saving protective power of human milk, about their mother's milk, about donor milk. And so that's when we should introduce the concept of necrotizing enterocolitis and why their baby, because they're born preterm, needs access to this potentially life-saving intervention, mother's milk and donor milk. I've said this, but I really want to emphasize it. Mother's own milk provides the most protection against necrotizing enterocolitis, followed by pasteurized donor human milk. It does not neither eliminate the risk of NEC. I really want to call out our nonprofit milk banks throughout North America who are helping to ensure that the infants who are in our neonatal intensive care units that may not have mother's milk, because there are a lot of barriers to mothers who are giving birth preterm, may not have enough or adequate supply to ensure their baby receives the milk that they need during this really critical time of development and growth. And so our nonprofit milk banks are key to ensuring that these babies receive the protection that they need from NEC. Parents are the most important member of the child's care team. And so when we're talking about engaging patient families, we really need to talk about how we can empower families, partner with families, center families as the most important member of their child's care team. They need to be treated like such at the bedside, in the unit, by everyone on the care team. The physicians, the nurses, social work, dietitians, everyone in the unit really needs to recognize that parents are the most important member of their child's care team and need to be valued and treated as such. And the way to prioritize our baby's health is by prioritizing the health of our mothers and our families. Mothers, healthy families are essential if we want to have healthy babies. And so we really need to take a holistic perspective and ensure that we're not just treating the symptoms of NEC, we're not just treating the infant in front of us, that we are recognizing that this baby comes from a family, that we are treating the family, and that we are caring for the family in a holistic way and showing up for the entire family. So I encourage you, if you are a bedside clinician, to empathize with parents, recognize the trauma that they are experiencing, recognize that these moments in the neonatal intensive care unit, it's going to live with them for the rest of their lives. And the words that you use, how you talk to them, what you do, will stay with them for a lifetime. Empower parents with information, with resources, with support, so that they can serve as an advocate for their child, so they can speak up and ask questions and share what's most important to them. What are they afraid of? What are they noticing that you cannot see? Because parents who are at the bedside are so tuned in to their child's needs and what may be going wrong. And so oftentimes the parents who are at the child's bedside hours every day, they're going to recognize when something is wrong before anyone else, before the monitors, before the clinicians. So we really need to empower families and empower the parents and give them the access to the information and resources that they need to show up for their child. We also need, again, if you're a bedside clinician, we need clinicians to anticipate what parents may need. And I remember that when I was, at my child's bedside in the NICU and PICU, I didn't always know what I would need in that moment or years off after that moment. And because, the clinicians are professionals and they do this day in and day out and they care for so many families, often for so many years, I think that they are oftentimes better able to anticipate what parents might need, not just in that moment, but years later. And so I encourage you all, as you're thinking about how to support families, how to partner with families, you could think about, well, what have other families shared? What can I learn from other families? What have I seen families ask for or need in these most difficult moments in the NICU and PICU? We definitely need to optimize the accessibility and the use and implementation of human milk in the NICU. We need every clinician, everyone in the unit to prioritize human milk as the life-saving intervention that it is. So again, the language that we use, how we talk to families about mother's milk, about donor milk, it makes a huge impact. We should be supporting mothers to pump at their child's bedside, making sure that mothers have access to a hospital-grade breast pump and the supplies, not just at the child's bedside, but at home. That should not be a barrier. We need high-quality hospital-grade breast pumps at home and at the bedside. We need these families to have access to peer-to-peer mentorship and lactation support, including on the weekends and in the evenings. It really needs to be ideally around the clock. And our families need access to healthy food and hydration so that their own basic needs are met so that they can then show up for their child and provide this life-saving intervention to them. So that's a bit about why I'm here, the urgency of NEC, the gaps in research, how we can partner with families. And now I want to move to what you all can do to help us build a world without NEC. The first thing I would ask, this is so appropriate because we're here with Medila, advocate for human milk. Locally, regionally, federally, in your unit, in your community, there are so many ways that you can help increase equitable access to human milk for all babies in the neonatal intensive care unit. One specific example I want to share is a partnership between the NEC Society and the UC Milk Bank in San Diego. We passed a law earlier this year that mandates insurance coverage for donor milk in our California NICUs for babies who don't have access to their mother's own milk. And it makes it easier for our hospitals in California to provide donor milk. So that's a really specific example of something that you could take if you're, in a different state in the United States. Take this legislation, take it and move it in your own state. There's lots of other regional opportunities. We have a NEC Awareness Day resolution that talks about the importance of human milk. So there's lots of ways for us to become and see ourselves as advocates for human milk for our babies. I also encourage you to utilize the NEC Society's resources for families who have been tragically diagnosed or bereaved by NEC. We actually have resource boxes in Medila. They are incredible partners to the NEC Society. Just spent the morning in their own office building these boxes to help ensure that all families that are touched by necrotizing enterocolitis don't feel isolated and alone like I did when Micah was diagnosed. When Micah was diagnosed with NEC, I had never heard of the disease. I knew no one who had been affected. There was no community. There was no collaboration. It was just the isolation and devastation of NEC. NEC is still devastating, but what we are able to change through partnerships with Medila and through these resource boxes, we are making sure that families don't experience that isolation, that they know that there is a community that is working tirelessly and relentlessly to build a world without NEC, that we are here for them, that we are here to honor their children and to do everything that we can to ensure that they are not alone and that other families don't continue to experience the devastation of this disease. These resource boxes are free to families. We do have academic centers and hospitals who will purchase them in bulk so that any child who is affected by NEC has access, their family has access to their boxes within their unit, but they're available on our website. And basically these have been built by us, our patient family team within the NEC Society, and it has information about what we wish we would have known when our child was diagnosed with NEC, when our family was tragically bereaved by NEC, and it's been reviewed by the NEC Society's Scientific Advisory Council, which is made up of the leading experts in the field of NEC. One of the things that we include in our bereavement boxes for families who tragically lose their child to necrotizing enterocolitis is a storybook called Forever Our Little One. And this is a book that I wrote, and my mom, who's holding Micah's photo there, illustrated. Because after Micah tragically died from NEC, I needed a storybook to read with his siblings that captured our grief, but also the joy of Micah's light and smiles and how we continue to share his light with the world. And so we created Forever Our Little One as a way for families to read it with the children they have in their arms, whether it's siblings, neighbors, if they're teachers, students. It really is to share with the people in their lives so that they can carry their little one and share their little one's light, who is with them always. So you can check that out on our website as well. The book is only available on necsociety.org. I think now more than ever, we need to be speaking up for babies and families. I mentioned a few slides ago that we should all be advocating for equitable access to human milk. Absolutely. We also just need to be, in general, advocating for our babies and our families and science, and to protect them from devastating cuts and threats that we are seeing today. A few examples of what we are doing and have done. We've passed NEC Awareness Day resolution in 9 states. It was just reintroduced in Congress a couple days ago. And so we would love to have your advocacy and your partnership and pushing these through. While right now it doesn't secure any additional dollars for us, what it does do is get more people recognizing the devastation and the urgency of necrotizing enterocolitis, so that we can move efforts like we did in California to ensure equitable access to donor milk, for example. And so these NEC Awareness Day resolutions are kind of a way to help expand advocacy efforts around awareness and around eventual securing policies and funding to help us prevent NEC. We also hope that you'll join us in encouraging your elected officials to prioritize and invest in science. The AAP neonatal team has an amazing campaign going on right now called Babies Need Science. If you're on Instagram, I encourage you to check it out. And it's true, our babies are desperately in need of science. You heard both Lars and myself talk about how there are gaps in research. And we right now cannot save all of the babies from necrotizing enterocolitis. The only thing that can save our babies from this disease is more science. And so we do need your partnership and your support to help push for advocacy to ensure that our research is being funded and supported so that we can save babies from this disease. Sharing information and facts about NEC, about the power of human milk, and inspiring your community to get involved. I am constantly inspired by how my community, the NEC Society's community, shows up for our babies and families. And it's incredibly moving and touching and powerful. And so if you are new to the NEC Society, I encourage you to join us and be part of this really powerful movement for babies and families. Just this last weekend on May 17th, which is Necrotizing Enterocolitis Awareness Day, we hosted a really powerful run. I live in Yolo County, and so we hosted the Yolo Run for a World Without NEC. And dozens of team members from Medela joined us, not just in Davis, where I live in California, but all over the world. Team Medela showed up to help us build a World Without NEC on NEC Awareness Day on May 17th. And so I encourage you, if you're interested in joining us, check out our website, check out how you can get involved in running, walking for a world without NEC. I also encourage you to join the NEC Society's community. We are a global, multidisciplinary, collaborative community that's dedicated to protecting the health of our babies and families. And so I encourage you to become part of this community. We have a research incubator. We have a nurse ambassador program. We are a tiny team, but we're working to expand how we serve the community and ensure that everyone feels like they belong here within the NEC Society, and there's a way for them to contribute to advancing NEC research, education, and advocacy. So I hope that you'll join us. As I mentioned, May 17th is NEC Awareness Day. The whole month of May is NEC Awareness Month. So I encourage you, it's not too late. Just by being on this webinar, you're helping to raise awareness about necrotizing enterocolitis. I encourage you to check out our website, see what events are happening, and to join really the global effort that's advancing NEC research, education, and advocacy. I would be so honored if you would consider joining us for the NEC Symposium in Chicago, September 7th through the 10th. This is being hosted in partnership with Cincinnati Children's and UNC Health with the NEC Society, of course. This is the world's largest conference dedicated to necrotizing enterocolitis. We hosted the first NEC Symposium in 2017 in partnership with UC Davis. And since then, we've hosted our NEC Symposium every other year. And so this is a very powerful, I would say, transformative meeting, because we don't just talk about the science, we talk about the real life impacts of necrotizing enterocolitis to ensure that our research is meaningful to the patient and families who are living through this devastation. And so not only will you hear from, at the NEC Symposium, the leading experts and scientists in the field, you will hear from real families who have been devastated by NEC, so that you can learn from their life experience. You can see what lessons they have to share to help propel research and to improve outcomes for our babies and our families. So I hope you'll join us in Chicago this September. And I want you to remember that, again, if you're a bedside clinician, families will always remember how you made them feel. the time that these families spend in the neonatal intensive care unit with their babies, it feels like it's going to go on forever, and it feels like it will never end, but it's a very short time. And during those moments that we're in the NICU with our babies, it's kind of ingrained in us. And the words that our clinicians share, the language that they use, it really touches us deeply. And we might not remember every single word, but we will certainly always remember for a lifetime how you made us feel in some of the most difficult, traumatizing moments of our life. So I think it's important to carry that with you. If you're a clinician, you have an incredible privilege and responsibility to help protect babies, help protect families, and recognize that even though we cannot eliminate the risk of NEC, we can ensure that no family feels further traumatized and further isolated alone during this time in the NICU. So show up with empathy, show up with care, and do your best to really invest in each family by building trust and rapport and taking the time to partner with families. And I just want to ask you to take one moment to think about what you can do to help build a world without NEC. It's helpful to write it down. You're muted. You could say it out loud to yourself. What are you going to do to help build a world without NEC? Because we can't do this alone. We need you. Our babies need you. Thank you so much. I am profoundly grateful to Medila for helping us build a world without necrotizing enterocolitis. I'm thankful to all of you for showing up, and just deeply grateful to everyone who makes the NEC Society possible. Thank you so much.
Wow, thank you to both Dr. Bodie and Jennifer for your presentations today. This was such an impactful webinar, and I'm very excited that we're going to move to our question and answer portion of the webinar. We've had a lot of great questions coming in, and again, please use the questions pane to continue asking. So the first question that came through today is, why is DSLNT found in some but not others? And how would a mother increase DSLNT in her breast milk?
Yeah, that's the magic question. First, to clarify, it's not found in some and not in others. It's found in some more than in others. So all women make DSLNT in their milk. It's just sometimes lower and sometimes higher. And that's, to a certain extent, a genetic variation that is normal. We see some associations with age. So if mom is a little bit older, the DSLNT concentrations seem to be a little bit lower. We haven't yet identified anything that we can change. Not much you can change about your age, unfortunately. So we haven't really found anything that is nutritional based or activity based or certain potential supplements or something like that you could say, okay, if I take this, it increases our DSLNT concentration. We don't have that data at this point, and we don't know exactly why it is higher in some than it is in others. But it's clearly an observation, and we see that it changes over the course of lactation. The numbers or the concentrations get lower as lactation stages advance.
Great, thank you so much. So as someone else asked, is testing of milk for HMOs likely to become affordable for milk banks in the near future?
Depends. I think if we really want it and we see that there is a need for it, we can make that happen at fairly low cost. It's just a question of who you bring on board, who develops certain technologies, and how we can scale it to make it affordable. So currently, we only have a few prototypes to test it. There's no commercial device at this point to measure DSLT concentrations at either the bedside or in a milk bank. With this, we currently don't have a lot of measuring the basics like proteins, fats, lactose, and total calories. And we're still kind of going in the blind there, and we pretend that all donor human milk is the same. And if we pool it, it will somehow even out, and sometimes we don't even pool it. I think there is, on the very basic level of understanding what donor human milk really is and how it varies in very basic components like the proteins and the lipids and the and calories, even there we have a huge gap still. So I think oligosaccharides is maybe the second tier of what we want to measure there. But if we think that this is important enough, it will happen. It will happen at a price that makes this possible. I mean, what is the price of wanting to make this possible? If you could save 1 or 2 lives saved from necrotizing enterocolitis, I think that price is not like, how do you measure this even? And certainly the technology is there to measure these things at point of care at a very low cost. We didn't spend millions of dollars to develop like small little devices. These are fairly affordable, and this is absolutely doable. We just need the force to want it.
Thank you so much, Dr. Bodie. So the next question is, what options do bereaved mothers have regarding their milk production?
Thank you. So I would love to just share that I am a bereaved milk donor. After Micah tragically passed away from NEC, I took his milk and I donated it to Himbana nonprofit milk banks. And it was a way for me to feel like I was sharing Micah's milk with other infants to save them from NEC. And so every mother and family is different. And everyone should do, obviously, what feels right and what is best for them. But I do strongly believe that all mothers and families should know that they can donate their child's milk after the devastating loss of their baby. Because I think too often what is assumed is that mothers just want to stop their milk production, and that it needs to stop as quickly and as soon as possible. And that people don't want to talk about it, that just shut it off. And I think a lot of mothers, not just me, don't agree. And what we want to be able to do is honor our child in the most powerful, amazing way. I think of it similar to an organ donation after the devastating loss of a loved one. Most people are familiar with the process of donating an organ to help someone else, save someone else's life after the tragic loss of a loved one. And I think that is true for donating bereaved milk as well. And so I would love to see everyone offering that to bereaved mothers. And what's so beautiful is that our Himbana nonprofit milk banks have developed, I would say most of them, if not all of them, have developed really beautiful ways to honor the bereaved mothers and families who donate their milk. And they honor the child who tragically lost their life, whose milk is now being donated. And so, of course, milk production can be stopped. I think too often it is stopped and not even giving the mothers a chance to learn about this really powerful way to save other infants and honor their child, and to connect with a community of other bereaved mothers.
I put a link in the here that links to the Cherry Blossom program that we have in our UC Health milk bank. So that's one example of how we support moms who lost their babies.
Thank you for sharing that. That's great. I know there's a lot of milk banks that are doing this as well. It's such a wonderful program. So our next question includes, what can milk banks do with regards to pooling milk and recruiting donors to increase the benefits in reducing NEC?
I started with that maybe. We've just from a research perspective, we've played this through. What happens if you pool donor milk? How many donors do you need to pull together to get above a certain threshold of, in this case, DSNT or other oligosaccharides? But also we looked at macronutrient content like proteins and fats and lactose, calories total. And I think it takes, if I remember the numbers correctly, 7 or 8 donors that we need to pool together to be safe when it comes to DSNT concentrations. The number is much lower when it comes to macronutrients. So there is research currently that looks into different pooling strategies. How many donors do we need? Ideally, this is all very blind, right? It's like, okay, let's grab a few milk bottles and then pool them together and hope for the best. If we put 7 or 8 different donors together, I think what would be better, and at least for macronutrients, we do that here in San Diego, where you measure the individual donors before, how much protein is in there, how much fat is in there. So you know what you're actually pooling together. You know, if you have a donor with lower levels and you pool that with a donor with higher levels, and you reach a certain average. So I think it's important to ideally screen individuals, make informed pooled decisions. And if you don't have that, then you're kind of fishing in the blind and you have to go higher in donor numbers that you pool together in one single. So ideally, information is always better than wild guesses. And I would always advocate for having technology that makes things in individual donors, and that informs your pooling strategies. But that comes with a price, of course, right? And I know that a lot of the milk banks are at a very, very tight budget. They are nonprofits after all, and they're trying to do this really on a shoestring. And they might not have the abilities to purchase equipment that's like $100,000 or something like that. So I urge the industry here to come up with ideas to create validated technologies that are affordable for milk banks to do these kinds of things.
I will also just share that I think there is a lot more we can do with empowering our community of mothers and families who have healthy babies, who have the opportunity to donate milk, to know that they can. Because I think a lot of people still are not familiar with the process of donating and what it means to be a milk donor and how it can actually save the life of an infant in the neonatal intensive care unit. I know that even when I had gave birth to my twins at 27 weeks gestation, I knew nothing about donor milk. I knew nothing about milk banks. And I think that's still true today for a lot of families, that they don't even know about donor milk. And if they have a milk supply and they have milk in their freezer, many families don't even know that they can actually donate this to nonprofit milk banks. And so I think in terms of a kind of a public awareness campaign, I think there's more that we can do to support families and to empower families and inform families of what does it mean to donate milk? How do you do it? And why is it urgently needed?
Thank you both for that. So a little bit more on donor human milk. How and when should parents learn about donor milk?
Well, I would love for actually this to be a conversation that happens maybe in high school or junior high, when our children are learning about the human body and sex ed and lactation. Like, why not just introduce it as early as possible? That's what I would say, is as early as possible. But that's kind of thinking really big, right? And so ideally, in my ideal world, people will grow up knowing about the life-saving power of human milk, normalizing breastfeeding, normalizing that this is how we feed babies, and it's potentially life-saving in the neonatal intensive care unit. My kids certainly know that, right? And so that would be in my ideal world, is for children to grow up knowing this. And so it's just kind of a given truth that our society and our culture, they recognize human milk. But since that's not the world and the reality that we live in today, I would say that certainly during when mothers and parents are expecting, I would love to see the conversation around donor milk happening then, right? So when they're going into their prenatal appointments and they're talking about perhaps, the birth and what happens after birth and lactation and milk production, then we can also talk about donor milk. And you can see kind of my theme here, right? It's not just about making sure families know about NEC as early as possible. It's really, how do we introduce the concept of milk as a life-saving intervention as early as possible, and the concept of donor milk and the concept of giving and donating donor milk for families who have the opportunity and privilege, right, to do this. And so I would love to see it as early as possible, certainly when families are expecting a child, certainly at birth, certainly at lactation consultant meetings, right? And so I think that's a great opportunity for lactation consultants as they're meeting with families and they're trying to get the babies to breast and to nurse, to also introduce the concept, hey, have you ever heard of donor milk? Do you know that there are nonprofit donor milk banks that, if you end up with an extra amount of milk in your freezer, you can actually donate it and you can save the life of an infant in the neonatal intensive care unit. That is powerful. And so as early as possible.
Can't emphasize this enough. Just to add to that, we always say this, right? It's not just about donor milk, it's about breastfeeding in general. We need to go back to normalized breastfeeding. It can't be that there's still this icky, oh, what's going on there? Breastfeeding. Let's cover up and do this and this. That just needs to stop. We need to be able to talk about breasts for what they're made for. They're there to make human milk, to nurture and nourish the next generation. And if we can't talk about this, and I fully agree with Jennifer, we should talk about this in school education. Biology classes, sex education, wherever you can talk about it, talk about the normal function of the mammary gland. What is it there for? It's so idolized and used for marketing and all kinds of stuff. but we completely forget what the normal function of the mammary gland is. And we need to get back to normalized breastfeeding. Absolutely. It can save lives clearly for the preterm infant when it comes to necrotizing enterocolitis, but there is so many other benefits of breastfeeding in general, not just for the baby, not just in the immediate context of breastfeeding while baby is breastfed, but for lifelong health, and also benefits for mom that we always don't think about, right? So we talk about, mimicking human milk and making it better, and making infant formula better, mimicking human milk. But the truth is, we're leaving out a completely part of the equation here, and that's maternal health. So highly emphasize what Jennifer just said.
I want to even go a step further. I also want to normalize pumping. So after Micah and Zachary were born at 27 weeks gestation, weighing 2 pounds, neither of them learned how to nurse at the breast. So even Zachary, Micah's twin, who came home and is beautiful and thriving and healthy in every way, even he, because of the NICU trauma he experienced, could not figure out how to nurse at the breast. And so I exclusively pumped for over 2 years. And so how do we not just normalize breastfeeding and nursing and donor milk, but also pumping? Because sometimes, given the trauma of the NICU, nursing and breastfeeding doesn't happen. We need to rely on breast pumps. Working parents, there's so many things that happen in life where we can't always nurse at the breast. And so how do we also normalize pumping? And I know it's getting better. We're getting to a world where there are more places to pump and the support for pumping parents. But we need to do even more, and we need to get people comfortable with it, normalizing it, and not making it weird or awkward. Like it is what it is. It's how we feed babies. And if my baby's not with me, I have this pump, and that's how I'm going to make sure that they get the life-saving, protective benefits of my milk that they need.
Well, both of your comments have truly resonated with the attendees. One of our attendees says that this sounds like a great opportunity for a commercial or Instagram, Jennifer. So definitely a topic that needs covered more. Excellent. So our next question, Dr. Bodie, do you know of a repository or any way for milk that has been left at a hospital due to discharge or bereavement that could be donated for research?
Yes, so we certainly have a biorepository here at UC San Diego. We call it Mommy's Milk Human Milk Biorepository. And we actually work very closely with our milk bank as well. So if milk is either expired or for whatever reason doesn't meet the criteria to make it into further downstream into donor human milk, then that milk goes in part to the biorepository, and then you'll be asked additional questions if you were willing to contribute to that, or it goes straight to our research labs where we either extract components out of the human milk to test out what they do in tissue culture or in other models. So we're making sure that every drop, even if it doesn't make it into donor milk, and a lot of it does, even if it doesn't make it in it, it is used for the biorepository or it is used for research purposes.
Excellent. Thank you so much for that. Someone asked Jennifer where the NEC Society currently operates from and what resources are available to give to mothers as far as education for preterm infants in the NICU and NEC.
Great question. So I personally live in Davis, California, so in Northern California, just outside of Sacramento. So we like to say that the NEC Society is headquartered here, but we actually do not have an office. I'm actually at home right now. And so I have a home office that I work in out of, and our staff team, our board members, everyone that is involved in the NEC Society works out of wherever they are in the world. And so we're spread out across the U.S. and globally. And the other question is what resources I believe that we have for mothers and families that have been touched by necrotizing enterocolitis. So we have the resource boxes that I mentioned on our website. We have a lot of educational materials on our website. We also have what we call the NEC Society Connects Facebook group. So it's a private, closed Facebook group that's only for families. So nurses, clinicians are not permitted to join. It's a closed private group just for patient families affected by NEC, where they can meet virtually online in a space that's created for them to share their stories, to share insight. And then we are working to also increase just the number of opportunities for in-person interaction between our families that have been affected by NEC. So a dream of mine that I have is to host a patient family advocacy summit to bring our patient families together in an in-person atmosphere where they can learn about how they can share their story to advocate for human milk, for the prevention of NEC, for a world without this disease. So, we are a small nonprofit, but we have big dreams. And, thanks to partnerships with Medela, and we are advancing our mission and getting to a place where we can better serve our community. But definitely check out our website.
Thank you so much for that, Jennifer, and for Dr. Bodie for sharing the link to the repository in the chat. So another question that came in, do you believe individualized or bio-matched donor milk will play a role in NEC prevention in the future? And if so, what will it take to get there?
I will let, I'm going to let Lars take that, but I guess I'll just say that I always like to start with mother's milk, right? And so talk about bioengineering and donor milk. Those are exciting conversations, and it's powerful and it's needed. But again, I just want to take us back to first starting with the mother parent's own milk, right? And how do we optimize that? How do we ensure that mothers and families are supported, empowered, have their hospital grade breast pump and their supplies at the bedside at home, that they have their basic needs met, that they have access to healthy food and hydration and ability to get to the hospital. And so like the bioengineering, that's an exciting conversation that I'll let Lars take. But I also just beg us to please remember that we need to start with our mothers and our families. And really, there's some simple things that doesn't take a whole bunch of extra technology, right, to develop. We don't need to there's plenty of research that needs to happen, but some of the most basic things we need to do is ensure that our mothers and our families have what they need so that they can show up and pump and supply their milk to their babies.
Absolutely agree, Jennifer. Sorry, I lost sound there for a second at the end of the question. That's why I didn't answer right away. So just to clarify this, the statistics that I showed before, 6 to 10-fold lower risk with human milk. So that tells you human milk is absolutely what we need to go for, right? That doesn't mean that it's 100% waterproof. There are still cases where we feed human milk and the baby still develops NEC. That's the tiny little fraction that is still high enough number. That's the fraction that we're trying to tease out right now. Why is that? And how can we use that information to get down to a world without NEC, right? So absolute bottom line, and I saw a few other questions. They're like, well, how do we tell mom that her milk might not be 100%? Your milk is better than anything else out there. That's number one. There's always a way to it's not a black and white thing, right? So how do we get to that last remaining risk? How do you identify what is potentially missing, or how can we bump something up, or what kind of technologies can we develop to get to that 100%? Will we ever be at 100%? Hopefully. Will always be a couple of cases probably that will escape that. But the more we understand about why human milk is so powerful and what this actually does, the more likely it is that we will reduce the numbers of NEC even further. If you're I don't want to say something wrong here right now. Use human milk, whether it's mom's own milk or donor human milk in your unit. If your unit is still not using mom's own milk or encouraging mom's own milk, or is still not using donor milk, please make sure that happens first. Then we can talk about how we can then get to the next step and improve the numbers even more. That would be my take on this.
Great, thank you so much. So we still have a couple questions left to go, but please keep adding them to the questions pane. Let's see. So there's been a lot of questions that ask about the impact of additives to human milk and how that may affect the risk of NEC. Is that something that you've studied, Dr. Bodie?
We haven't in our labs. I saw there are a few questions about fortifiers and whether it's human or bovine, other things added to it. I think the data is not particularly strong that it has to be human over bovine from what I'm recalling from the literature. I think the type of fortifier is not as important as actually giving human milk, independent of what the fortifier is. So that would be one of those answers. And what else is added to it and how the milk is treated. I can say, at least from the oligosaccharide perspective, that current Holter pasteurization and other treatment methods to reduce pathogens are actually keeping oligosaccharides intact. So that's a plus. And that tells us that oligosaccharides are indeed part of the solution here, because we know that some of the bioactive proteins, for example, get destroyed during the pasteurization process, and yet still donor human milk protects against NEC. So that gives me some confidence that oligosaccharides really play a role here, because they remain intact and the protective potential of donor human milk is still there. That's all I can say to it. Jennifer, you have any other?
Yeah, so I think this is an important question, and it always has a lot of interest from the community. And so there's a few things that, because I was reading quickly through the questions in the comments, and so I will just personally answer. I'm very comfortable and open and sharing. Neither of my babies received formula. They received my milk, and they did receive a human milk fortifier. I do think it is absolutely critical for us to be very clear with families what is a human milk fortifier? Because I'll say that when I was in the neonatal intensive care unit with my babies, when they said we're adding a human milk fortifier, I imagined it was like a fortified like cereal, right? Like where, where you fortify cereal, you just add some like vitamins and minerals, and there it is. It's the same cereal, just with added. And when I realized they added the human milk fortifier, it was bovine. And I asked, well, what are you adding? Like, because I noticed a change in my baby's stool, and I was like, what is this that you're adding? And they brought me the bottle, and then I realized what it was. And Lars is correct. The evidence is inconclusive. We need more research on the fortifiers. But certainly what does need to happen is ensuring families understand and know and empowered with information. And because I think the term human milk fortifier is pretty vague and unclear to most families, it certainly was to me, of what it is and why it's being added. And so I think it is important to just communicate with families. Why do we feel your baby needs this? What does it do? How does it help? What are your options? I will say that in the US, most babies are absolutely just fortified to ensure that they have adequate growth and nutrition, but that's not true for the rest of the world. And so the variability is pretty wide in terms of who and where and what fortifiers are being used if you look outside of practices, outside the US is what I mean. So I think it's a really good question. It's an important conversation to have. There is not a clear black and white, right or wrong answer right now. But I think that empowering families, having conversations with families, making sure that they understand why a certain product is being utilized and implemented for their child's nutritional needs. Parents need to understand and know why something is being added to the milk. And that way they're invested in the decision. They are able to ask questions and also be comfortable with what is taking place. So yeah, I just think that conversations and transparency are really important.
That's such a great point, Jennifer, really having an informed decision with parents involved in the neonatal intensive care unit, such an important thing as a nurse that I would say was great to have the parents at the bedside and part of those conversations. Excellent. So we have just a few minutes left in the webinar. I believe we have covered all of the questions that have come through the questions pane. So I'll leave it for just another moment here before we conclude to see if any others come in. But I'll also give you both an opportunity. Is there anything in closing that you'd like to say? This has been such a great, engaging webinar. Thank you, everyone, for participating.
Thank you. I would just say that I continue to just be inspired and filled with hope that we can, within our lifetime, build that world without NEC. And I really do mean a world without NEC. I know we have a lot of global attendees on today. And even though the NEC Society is a tiny little nonprofit that it happens to be based in the United States, we do, as best as we can, take a global international approach to the problem of necrotizing enterocolitis. And we are, even though we're so small and our resources are so limited, we are eager and dedicated to ensuring that we are doing our best to serve the global international community and raising awareness and increasing research, education, and advocacy for NEC babies and families around the world. And so I'm just really grateful and inspired with hope and gratitude for our partnership with Medela, and to all of you for helping to increase equitable access to human milk, to donor milk, because that is really the best thing that we have, right? We need to prevent preterm birth, but until we can prevent preterm birth, the best, most powerful intervention that we have to protect our babies from NEC is human milk, mother's milk, donor milk. So just thank you to everyone who's helping us advance that research, advance that advocacy, and give more milk to more babies.
Thank you, Jennifer.
And I say small but mighty, Jennifer. This is not small. It might be small in numbers, but certainly not in might. I'm always amazed. I've heard you speak multiple times, and I know you for a few years now. Your passion is really moving here and is moving mountains, not just small needles. And it's amazing what a single person based out of your passion and your experience can do to move research, clinical advocacy, everything that you've been involved in is absolutely remarkable. So kudos to you. This is not a small thing. This is a huge thing. And if there were advocates for diseases just like yourself, we would hopefully have a much healthier world today. And I just like to thank you for your partnership, Jennifer, with the NEC Society, everything that you've done there. Absolutely. Medela for their support again, but specifically for this topic and partnering up with the NEC Society, also being a partner for the Human Milk Institute. I think that one word on that slide that says building a world without NEC is the word together. Together we can build a world without NEC, and that includes everyone who's on this call, and everyone's on the webinar, everyone who's on the NEC Society, everyone who's doing research, the clinicians, the nurses, the companies like Medela, the nonprofits like the milk banks. Together we can make a difference here. And that's a little bit lost in today's society and today's climate, that everyone's like always finding what's the difference between me and the others and how I can get ahead of something and be different from other people. That's not how it works. That's not how usually things are moved. We're moving things together. And with the current climate, with the NIH funding and the whole idea about research not being so important, you've seen how long it's taking to get something like our discoveries into actual clinical practice, and we're still not fully there. It's taking too long. It can't be that it takes 17, 18 years to get from a first discovery over to clinical practice. And this whole idea of defunding NIH and NSF and not investing into research, that's going to make us way more sick than we are already, and is not going to help us get healthier. So I hope that everyone advocates and votes the right way to make sure that research gets the funding, that clinical endeavors gets the funding, that some of the foundations that are also under fire get the right status and funding, so that we can continue together to make a difference here, because we are under attack, and that is not the right time to cut on science and clinical work and whatever is cut there. So thank you.
Yes,
and our babies are counting on us. Right? Our babies are counting on us to do everything that Lars just said. There's no one else that's going to do this work. Like, we are the ones. And so we all do need to step up. We need to protect our babies, need to protect our families, need to protect science so that they can have healthier lives, that they can be protected from these devastating diseases like NEC. Our babies are counting on us to do that.
That's such an important point you've both talked about. And there's been a couple additional questions in the chat, Jennifer, about how are you going to continue to move forward in your journey with the NEC Society, given that there's financial instability and cuts to funding and grants?
That's such a great question. And I'll tell you, the last few months, I was really nervous. We have received NIH funding previously, and we put together a very compelling, I thought, beautiful application to the National Institute of Child Health and Human Development to support the NEC Symposium, which we've been successful with previously. And that was not even acknowledged or considered. It was flat out rejected without them even really taking a look at it. And so when that happened just a couple months ago, I was very nervous, but I am now filled with hope, because right now it's NEC Awareness Month, and I'm seeing people and partners and groups and companies step up in a way that I've never before. And so even when there are politicians and people attacking science and attacking federal funding and resources that we've relied on before, there are other people that are stepping up to fill the voids and fill the gaps and take care of each other. And that's what I think we really need to do, and to lean in on, and to recognize that despite the attacks that are happening, despite the threats, that our community is solid and sound, and that we have each other, and that we can continue to lean in on each other, rely on one another, collaborate, and partner. And that's how we will continue to serve our community and our babies and our families.
That's such a great way to close the webinar, Jennifer. Thank you so much. And so thank you both, Dr. Bodie and Jennifer, for your insightful contributions today. And thank you everyone for joining us in the webinar, Advancing the Science and Accessibility of Human Milk for a World Without NEC. We're so excited that both Dr. Bodie and Jennifer have highlighted their upcoming symposiums, the Human Milk Institute and the NEC Society, and we really hope that you'll consider attending both of these important events. And additionally, I'd like to personally invite you to Medila's Breastfeeding and Lactation Symposium Series, the Americas edition, which is taking place September 11th and 12th this year. Registration details are available in the handout section of the webinar today and through the link that I am going to be sharing right now. And I'm also going to post it here in our chat. So as we conclude, you're going to be redirected to a brief survey. Your feedback is truly incredibly invaluable to us, and we really appreciate that you take a few minutes to share your thoughts. A follow-up email will also be sent with a link to the webinar recording for your reference. And so on behalf of Medila's Medical Affairs team and our presenters, thank you again for being with us today, and we wish you a wonderful rest of your day. Thank you both.